It's divided into two buildings. The main one is in the northern part of the city, and it cares mostly for MSM. And it's situated in a middle-class neighborhood in Mexico City. It also has a transgender clinic that's very important in the country. And the other one is a newer clinic that was built about 3 or 4 years ago, and it's situated in one of the poorest neighborhoods in Mexico City. It's a very densely populated neighborhood, and it's also associated with a lot of poverty, drug use, teenage pregnancy, low education, etc. And so we decided to collaborate with this clinic. And so we're now doing HIV genotyping for everyone that is diagnosed in this clinic. For you to get an idea of what this clinic represents for the epidemic in Mexico, I have some statistics here. So these are the number of people who are detected at the Condesa clinics in Mexico. And as you can see, this clinic is a key detection center in the country. In 2016, they performed 31,000 tests, and from them 12% were positive. And from these, most of the people who get tested are men, especially MSM. And as you can see here, the ratio of males and females who are diagnosed at the clinic is widening. This is worrying because it means that every time less women are getting tested and more men are getting tested. And also, right now, the number of people who are enrolled at Clínica Condesa are about 13,000 patients. So this clinic alone cares for 13,000 patients in the country. And the other persons that are diagnosed in Clínica Condesa go to other centers and to get enrolled in treatment. And so we thought, well, this is a very important center for the epidemic in Mexico, and it's a good place for a proof of concept study on implementing HIV drug resistance testing for everyone in the country. So we're performing baseline genotyping for all the persons that are diagnosed in the clinic and accept to participate in the protocol. And also, we are trying to combine molecular data and epidemiologic information to help understand HIV transmission dynamics and to design directed public health policies to tackle specific sites or populations at higher risk. And we also think that molecular and epidemiological information could be used to obtain real-time data to plan interventions to detect hotspots of HIV transmission within the city. So right now, we're starting to collect behavioral and epidemiological data for all the participants in the study. And these are some preliminary results for this study. Right now, we have enrolled more than 1,400 persons, and this is just the pretreatment drug resistance prevalence in this population. We are, as you can see, the NNRTI drug resistance is reaching the 10% threshold. It's very similar to the national average. Also here you can see that efavirenz and nevirapine are the drugs that are mostly affected by transmitted drug resistance, specifically because of the high prevalence of the K103 mutation. That's the highest bar in the lower graph. And that's to be expected. That's also been seen in other countries in the world. This mutation can be easily transmitted from one virus to another. And also an interesting thing is that resistance to integrase inhibitors, as you see in the blue bar in the graph on the left, is very low still because these drugs are almost not used in Mexico. And this is very different to the scenario in the United States where they are already using integrase inhibitors as first-line regimens. And also with the help of Antoine, we have done this transmission network for all the patients from Condesa Clinic. Here you see the network, and the nodes that are colored are persons who have transmitted drug resistance. The ones in red have NNRTI, transmitted drug resistance, and some of them are marked. Those are the ones that have the K103 mutation. That was a mutation that was most frequently transmitted in the network. In general, 31% of the sequences were in clusters. We found 295 sequences clustered, and the number of clusters was 21, and 18% of the sequences within clusters have transmitted drug resistance. The most frequently transmitted mutations were, as we have already mentioned, K103N, but also T215S, Y188L, and M46L. And an interesting thing is that we did our sequencing with next generation sequencing, and with this technique you are able to find HIV variants that are at low frequency within each person. So you can detect variants that are over 2% of the whole viral population within each person. So we also wanted to see if these variants that can be present at low frequency can be transmitted from one person to another, and we see that that's not the case, at least in this network. The persons who have variants at drug-resistant variants at low frequency are shown with a green perimeter. And as you can see, none of these persons transmitted the resistant virus to their partners, and this is important to know. Right now we're collecting the behavioral and data for all these persons, and soon we're going to be able to show what are the risk factors for transmitting drug resistance in these networks. So our key messages are that we are seeing an increasing amount of new diagnoses among MSM in Mexico City. Also, pretreatment drug resistance to NNRTIs. In all persons who initiate antiretroviral treatment at Clínica Condesa have more or less similar levels to those observed at the national level. Our network analysis demonstrated frequent cases of shared drug resistance mutations among linked individuals, especially the K103 mutations. And so we know that pretreatment drug resistance can be spread from one person to another. We did not see any cases of transmission of low-frequency drug resistance mutations, and we need to strengthen retention in care, since there is a possible role of chronic intermittent users as conveyors of HIV drug resistance. Many persons included in these networks were prior users of transmitted drug resistance, and maybe a way to stop people from keeping or transmitting the resistant virus is to keep them in care, to link them in care. Also, there's a need to obtain and rapidly deliver HIV drug resistance result to treating clinicians. That's a challenge in our setting, because we have so many sequences that are delivered to us that delivering them on time is difficult. And also, using networks could help find clusters as well as specific venues or locations to direct HIV prevention in Mexico. And that's, I think, we're working right now with Antoine, with Sanjay and Joel. So we are trying to implement this real-time detection and of hotspots, or of increasing clusters to be able to act upon them. And I just want to thank all the persons involved in this work, especially Dr. Gustavo Reyes-Terán, that's the head of our center, people from Censida, that's our national program, the Clínica Condesa, of course. Andrea González is the head of Condesa Clinic. and the people from the WHO, people from Universidad del Valle de Guatemala, who are helping with the Central America studies for the HIV drug resistance surveys, and finally our funding sources. And thank you.
It's a neat talk. I really like the insight about minority transmission variants. That's quite nice. You painted the picture of sort of NNRTIs versus integrase inhibitors, but there's also the NRTI route, and the only NRTI transmission you saw in that network looked to be a revertant mutation, 215S. So would this argue in favor of at least transitioning away from the NNRTIs towards an NRTI before integrase inhibitors become affordable?
I don't think that's the case. So still the NRTI resistance levels are less than 5% in our setting. These results very much reflect the national data. So as you can see, the NRTI prevalence is about 4% here, and is more or less so at the national level. So we are still confident that we can use a first-line regimen based on the classic 2N-RTI backbone. But including dolutegravir would be very useful because of the high genetic barrier of dolutegravir, so we will stop drug resistance from generating using these regimens. That's a problem when using albitegravir, for example, or raltegravir, that have very low genetic barriers, and would be a problem if mutations exist in the NRTI backbone.
That's really nice and interesting data. I just have one comment about the NRTI mutations. These small number of AZT-associated mutations, as well as the M184V mutation, have been shown not to impair the treatment efficacy of tenofovir-based regimens. So I have a couple of very practical questions regarding the implications of your data. One is, are these provided to make treatment decisions for the initiation of treatment for these patients?
Right now, as I told you, we have been discussing this with a national program. So right now, there are no official recommendations yet. But one of the other things that the national program recommended was to repeat the national survey having sub-regional representativity. So we divided the country into 8 regions. We're performing the survey with regional representativity in each one of the 8 regions. We're going to have the results by the end of this month. And the idea is that Mexico is very diverse and a very complex country, so we would find differences within the different regions. And so maybe acting at the national level would be difficult, but we can start with the regions that are most affected by transmitted drug resistance. Right now, with preliminary results, we're seeing that the southern part is more affected by transmitted drug resistance, especially the states of Oaxaca, Guerrero, and Chiapas, that are the poorest states in Mexico. NNRTI transmitted drug resistance is very high there, and maybe we can start with those or prioritize those states, and then continue with the rest of the country, while we get a good negotiation of prices for dolutegravir.
Right. So it's clearly been shown that NNRTI resistance predisposes to higher rates of treatment failure. So
yes,
it's known that will have an impact. Regarding the alternatives, it's either PIs or integrase inhibitors. The current single pill formulation of tenofovir, lamivudine, and dolutegravir in sub-Saharan Africa is $75 for a year.
It's a big difference.
So I'm sort of curious what the costs for both the PI alternative and the integrase alternative is in Mexico.
So the combination, the one pill combination of albitegravir with tenofovir and emtricitabine is similar to a tripla. So that's why the national program recommended to think about that option. The single pill combination of dolutegravir, abacavir, and lamivudine is still not included in the basic list of drugs in Mexico, and we need to have it approved by our kind of FDA. So it's called COFEPRIS. They need to evaluate it and accept it first, and then they have to negotiate the prices so they can be at similar places, similar levels as a tripla. So there's still a long way to go for drug negotiations to work. And the other thing is to push the use of dolutegravir and have the price reduced, because dolutegravir as a single drug is already approved by our COFEPRIS, but the triple combination is still not approved. So it's complicated in the bureaucracy and the pharmaceutical company interests and even the people's interests in this. There's a lot of money involved here. But anyway, we need to get there. And this data are showing that we need to do something, and at least the national program is already aware of this data, and they're planning to act upon this.
Yeah, sorry.
Okay, one last question.
Yeah, sorry. Do you ever test for acute HIV infection in these programs that you're looking at?
At Clínica Condesa, they do it, but sometimes they run out of reagents. It's terrible, but they're doing fourth generation ELISAs, Western blots, and viral loads for detecting acute infections, especially the absence of P31, the P31 band, and the Clínica Condesa is a good center to find acute persons. In general, people in Mexico arrive to clinical care very late in the infection, so most people are detected late. But in the country, that at the national perspective, the Clínica Condesa would be the one that finds the most or the highest proportion of people in the acute infection.
Yeah, I think it looks like it's hitting the right population and that you might be able to access it pretty well.
Yes.
And then Susan Little, raise your hand, has an acute infection program that's looking at trying to export it to other places. So this might be a good connection.
Yeah, I think this would be, if you're planning to work with Mexico, this would be the clinic to work with, because they are the ones that have the highest proportion of acute persons. Thank you.
Thank you.